this will end up being a long video. I may break it up into two, but here we're going to talk more about the promoter region. And we have a graphic here, and of course we understand our gene here. This is your terminator down here, so there would be a hairpin loop. Alright, here. And where is our transcription start site? That would be TSS right here. Now realize we are in DNA. I'm going to have you do some analysis. The sequence I gave you, is it DNA or is it RNA? Keep that in mind. You may not have done exactly what you needed to do in previous ones because it may not be clear if you have DNA or RNA. And then of course you have to worry about, is BLAST sending me DNA or RNA? And I'll tell you, BLAST will send you both. If you just don't tell it anything, it will give you both DNA results and RNA results. So but what are we looking at in the sequence I gave you? Because I don't believe I told you if it were DNA and RNA. Now, if it's genomic, you assume that it is DNA, not RNA. But, and you can't tell with a FASTA. But we've already seen that that's confusing, and they sometimes mislabel it or just say genomic and it's really RNA. So be very careful. And I'm going to have you go back and check that in another part of the project where you everything as if, did we get this right? And you'll go and check yourself. But we won't do that now. We'll just, however you've been doing it, continue to do it the way you're doing it. But keep that in mind here as we move forward. Now, in this section, what we want to learn in this section is just where is the promoter region. That's the first thing we need to figure out. The regulatory region right here. That is the promoter region. It's the region where you have all the things that regulate this gene. Do you splice? How do you splice it? That's one type of regulation. How many do you make? Alright, do you turn it on and off? So that also means that this is the region that you're going to get your C's and G's. So you're going to get CPG island. Do it this way. CPGs, alright, is if you're going to have one. Now you don't always have one. Alright, and then, oops, let me go back. Alright, now in the case of flies, they use different letters than C's and G's, so it's much more complicated in flies if you ever study flies. But in vertebrates, you usually get CPG islands, and in plants, you usually, insects went off and went somewhere else with it, which is rather interesting. Now, not all genes have CPG islands, and we'll talk about that in this lecture, which ones are likely to have them and not. So we're focusing, since we used CPGs to find the genes, we're going to use the ones that are CPG islands. So you can be assured that these genes are the type that have CPG islands. Alright, now moving on, where do you find things? So the transcription start site, this is where the polymerase is going to start copying. So in other words, you're going to start making your RNA here. Alright, you're going to start making the RNA right here. That's your transcription start site, and we call that +1. Now, that means that is not where you start translating it into a protein. You start translating it into a protein here at the start codon, translation start, so they're saying that's here. And you stop at the stop codon, and that's here. So a lot of you have been confused this semester about, you get transcription start, where is this? Is it RNA, DNA? Well, the start is in the AUG, alright, or ATG in a FASTA, is in the RNA, alright, and the stop codon is in the RNA, but you can read it here. If it's ATG here, if you're doing DNA, ATG, then if you go backwards, that's a TAC. So if you're in DNA, you could stay in DNA and go, this is TAC. If you're in RNA, well, you have to go backwards. You have to keep track in your sequence. Am I in RNA or am I in DNA? So I just want to make that clear that I may not have been, you don't know, I don't think I told you if you have DNA or RNA. Now, if it is a true genomic sequence that we just went out and sequenced, if it's probably in DNA, and you would think that, but go check, make sure you're doing everything the way you should, because if you're going to find, if you have DNA, then you need to, find the complement, all right, here to find the start and stop codons. All right, so you may have not found the right start and stop codons. Now, where do you find your transcription start site? Well, that's in DNA. All right, the only part that's in RNA are the codons. All right, so RNA really, you really only look in the codons for RNA. All right, and this is difficult to keep track in your head. I know. When I was an undergraduate, this confused me so badly, but you just have to work with it long enough and you'll figure it out. It's just, you got to remember, am I in DNA or am I in RNA? And if I am, what am I looking for? So codons, all right, so if something is a start codon or stop codon, you're in RNA. Otherwise, you need to be looking in the DNA. Now, CPG islands, even if you have DNA, even if you're looking in RNA, you can find the CPG islands in an RNA, because you can, if it's a bunch of C's and G's next to each other, they will translate into something very similar. And in fact, I'll tell you, usually it's easier to find a CPG island by looking at the RNA, because it reduces all the clutter, I guess. There are too many of them, all right? And sometimes you can go and use the RNA if you find the complement. It's actually sometimes easier to visualize the CPG island if you do switch to it. So, but the reason that it doesn't matter with CPGs is because C's and G's translate to G's and C's, and since you get a bunch of them together, you usually end up with the complement being showing you where there are a lot of them. So if you have a lot of C's and G's, you're also going to have a lot of CG. In other words, like this. So if this is your DNA, this would be your RNA. And as you can see, you end up with a CG here. You end up with 2 up here. And so when you actually look for CPG islands in an RNA, if you have that region, realize I shouldn't say RNA if you have it in the complement, because you should never have an RNA with CGs in it. You generally, the CGs don't make it over into the gene. But if you did have the complement here instead, if it's given to you, which they do to you in the database, then you would you just eliminate a little bit of the clutter, but it actually really sticks out more when you do it this way. But it doesn't matter. I will let you guys, that will be a different part of the assignment that we will come to later. So what I want to show you, though, is the transcription start sites. Now, where is the UTR? The UTR is this region right in here, and that would be the 5' UTR. Right here they're showing you. Yes, you can have binding sites for transcription factors. Proteins can bind in there, and in fact they go into the first exon. So in other words, this whole region from, say midway through the first exon all the way back, and tell you the truth, enhancers can be 100,000 base pairs. They can be on the other side of other genes. So this distal promoter, this is what we were going to call the distal promoter, there can be genes in between the proximal promoter and the distal. So you might have, this might be like way over here, okay? You might have a couple of genes in here, and these enhancers kind of remind you of maybe an operon where you have several genes being controlled, and in a way enhancers do work that way, and they can be really far away, at least some of them. Some of them are going to be in here. Some of these these transcription factors are going to bind in here. Now the proximal promoter, this is the one that is a little not that far away, and definitely not, there won't be a gene in between here and the proximal promoter. The proximal promoter, you get these bending proteins like the GAF or GRAF, and we're going to talk about that. And your TATA-box, things like that we have talked about, they're going to be much closer, and they're closer to we're going to call those the core promoter, though really the TATA-box is not part of the core promoter. The TATA-box is there to make sure your gene is transcribed in only one direction. In other words, you have two strands, and we're going to talk about that here in a little bit. And RNA polymerase can stick, but does it go this way? All right, or this way? All right, it can work. You can have two genes kind of back to back here on different strands, and the TATA-box just makes sure it goes one particular way. And you know, it's a lot of the problems we have with the literature in this and the way we look at it is due to the and the way they teach it in introductory books versus what's really going on, is that we got it wrong early on. We assumed a lot of things. One, we assumed that eukaryotes work like prokaryotes. A lot of mistakes were made. And so the stuff you learn in a Biology 101 book in many ways is not true. And so you have to keep track of where everything is and know what's possible and what everything does as well as we know. We don't know a lot. We do know now that RNA polymerase will just come along and stick anywhere. It really doesn't matter. So RNA polymerase, oops, didn't mean to do that. There we go. All right, RNA polymerase can stick. Oh, it's not drawing. There we go. I've lost my drawing ability. No, I haven't. Doesn't like drawing on the picture? I don't know. Anyhow, so it can stick here. It can stick. See if I click there, it can click there. You know, RNA polymerase can stick all along the DNA here, but when it sticks, does it stay, and can it move forward? Well, a lot of the times it can only move forward in one direction, because it can move. It can stick on either side, or it's dual. When it sticks, it can move in either direction when it sticks, and if it moves in one direction, the TATA-box will be there to tell it, no, don't go this way, go the other way. All right, and that's what the TATA-box is for. We know that now, but is that what it's for in the prokaryote? Maybe, and actually that's something we're not sure. We know that this is kind of reverse. We know that's what it's for in eukaryotes. We're still not sure, but it looks a lot like a TATA-box. It's assumed that they evolved from one another, but maybe they weren't, because we're not sure exactly how the prokaryote TATA-box restricts direction, and even because you don't get as many. Well, you do. It's like I said. We don't know. There are still many things we don't know. So keep that in mind. So the normal TATA is not really where it sticks. It just sticks near TATA. All right, put it that way. And at least in eukaryotes. All right, so I'm going to move forward here. We're going to talk about some things. So here's your promoter definition. And your promoter definition is essentially the regulatory regions overlapping a transcription start site. So this is kind of an old one, but here's your transcription start site. And so one of the definitions is this one, and it says that the area around the transcription start site is the promoter. That's a little bit of an old definition. We now kind of go, there's the core promoter, and this is more of a definition of the core promoter. All right, and the core promoter is this region where RNA polymerase sticks. But then there's this proximal promoter, which is nearby. All right, then you get the distal promoter. Now, the proximal promoter will include the core promoter. So if this is the core promoter, I should say, if this is the proximal promoter, the core promoter is this area in here near the TATA box and before the transcription start site. Remember, RNA polymerase will stick before the transcription start site, and in eukaryotes, the TATA box is about 30 bases down, not 10 or 11 like in prokaryotes. So it's much further away, and this region in here is called the core promoter. And just remember that they are different between prokaryotes and eukaryotes, and maybe it's something you would learn in molecular. I don't know where Dr. Houston went with that, but just realize that it is not the same in eukaryotes and prokaryotes. Alright, so just keep that in mind, and this is kind of an old definition. Probably a better region for everything we're doing here is what they say here, a regulatory region. And we have broken this regulatory region into the core promoter, which is inside the proximal promoter, and then the distal promoter, which can be way, way, way out here. Alright, so the distances here we'll talk about. They're different in different species and in different types of genes. So the size or the length of the regulatory regions change depending on what's going on, but we'll have general outline guidelines for you here. Alright, so moving forward, the core promoter, okay, here is a better idea. The core promoter is generally defined to be the stretch of DNA that directs the initiation of transcription. The core promoter includes the transcription start site and the surrounding sequence that attracts the pre-initiation complex. In other words, it attracts RNA polymerase. Alright, there are some other proteins that get attracted first, and then RNA polymerase is added on top. So let me draw that. So here you have, let's say, your transcription start site. Alright, and let's go ahead and give ourselves a TATA down here somewhere. RNA polymerase will bind, somewhere, like right in here. The TATA is a backstop to make sure you don't go this way, on this strand. So in other words, you could be on the other strand, and when RNA polymerase binds, it could go that way, but the TATA is here, so it makes sure that it goes this way. And so this is your core promoter. It typically does include the TATA, alright, and it cleans all the way up to the transcription start site. And these are where proteins, all kinds of proteins, are going to stick here first, and that is the pre-initiation complex, PIC. I'll never ask that probably. That's more of a molecular biology question. You can take that in molecular. And then RNA polymerase will actually stick on top of these proteins, and once it sticks, then you're ready to go and copy the DNA into RNA. Alright, so let's see, what did we... No, we're good. Alright, so core, promoter, proximal, and distal. Alright, so eukaryotic promoters are much more complex than those found in prokaryotes. Yes, and we do not know everything. The promoter region is broken down into two regions, the proximal and the core. And here we're going to say 300 bases before and after the transcription start site. So that's a pretty good length, and I can tell you that that changes depending upon species. This is not cut in stone, but we'll work with this one. So this is something I want somebody to find. So as you move forward, someone agree that they will go find this, and they'll define it. Now, this is also a region that you are probably going to have your CPG islands. So the proximal promoter is going to have CPGs in it if you have a CPG island. So is this as far back? No, you can. In fact, I think a better definition of the proximal promoter is, if you have one, it's where your CPG island is. I think that would be a good definition. So if the region where you get more C's and G's versus another region, they have it as 300 bases, but I'll tell you, if you count it as a CPG island, that defines it, then it might extend to 500 or 600. But it usually doesn't go past, I can't ever remember it going even to the end of the first exon. So maybe it did. Maybe I have seen that. Yeah, I think I did. I saw it go through the first exon in one and into the first intron, barely. But, so I guess I have seen that. So, but generally 3 to 600 base pair. It could be much smaller too. But you can generally think of the proximal promoter as the region where the high CG, where the CPG island is. All right, and that's a good way to define it. There are no set definitions because we're learning and it changes, and they will come up with something to put into a textbook, but it's there just to make it easy to write a textbook. It's not because that is really what's going on. Now then, the distal promoter contains enhancers, silencers, and insulators. Now these are not transcription factor binding sites necessarily. These are things that do enhance transcription, like you make more of something or less. These are things that can insulators block enhancers and silencers. Silencers decrease transcription. So this is if something binds to the distal promoter that increases it, we call it an enhancer. If something binds to the distal promoter and it lowers it, we call it a silencer. And if an insulator will be something that negates one of these. All right, so an insulator will make sure that these don't affect it. So if something binds to an insulator, then if these are present and something is binding here to cause this will negate it. So you need to know the definitions of an enhancer, a silencer, and an insulator. An insulator just says, hey, ignore these if they are present. All right, that's what an insulator does. But you have to have a protein binding there. So if I had each of these, an enhancer, a silencer, and an insulator sequence that proteins bind to, so we'll say these proteins bind to it, well, if it were just this one, you would make more RNA. If it were just this one, you would make less RNA. All right. If you have both of them, who knows what happens. But if you have this one, you can ignore these completely. All right. Think of it that way. Now, the distal or pre-promoter, like I said, this is the region that you usually get these things that act from a distance, and they may act on multiple genes at the same time. So realize this is more like the operon where you had Z, Y, and A, but don't think of it that way. Remember, these are separate genes regulated separately in their own individual promoters. But then there are larger regulatory structures further away, the distal promoter, which may be working on several genes at one time. So it's similar to the promoter near it in that it can regulate it, but then it's another level which when genes need to be regulated as a group, like in an operon, you could regulate the several genes at once with enhancers, silencers, and insulators. All right, but I will get to this in a second. All right, so here's how this actually works. So here's your promoter, here's your gene, and then you might have a couple of genes in here between it, and then here are your enhancers. And here's your enhancer with the, or I should say, maybe they should call this instead the distal promoter, or further down. That would probably be a better way to call this. This is, like I said, the terminology is really hard to work through in papers because different scientists will use different terms. There's no set. So this is the distal promoter. All right. And you have these bind, and they may activate all of these, but you might have something in here that will negate this. So this insulator might actually be in here, and if that binds, that would negate whatever these were telling you, like increase or decrease or something complicated. But a lot of times these silencers will be like Gaga. What they're doing is they're not bending the DNA correctly, and when you don't bend the DNA like this correctly, these things don't bend over and enhance it, and therefore you don't get as good of transcription or you weaken it. So a lot of times something like a GAGA will be this protein that sticks here that allows it to bend over. And that's what's happening here. This piece is bending over to come and attach here. So all these things that are attaching in here are actually up here when they bend over, all right, and they are attaching above the core promoter region, and they attach, and depending on what's stuck up here, you either speed this up or slow it down. And then of course if there's something in between here that sticks and says, oh, I'm not going to let you bend, well, that would basically silence, inhibit these. So we go back, I need to make sure we understand. That would insulate, that's the correct term to use here. So that would be, in this case, something that wouldn't let it bend would be an insulator. Something that causes it to bend would be what we would generally call your standard transcription factors. All of these at different times have been called different things. In general, we say any of these sequences are transcription factor binding sites. And again, the literature is horrible in defining these. There's no big body that comes together in this and says, this is what we're going to call these things. And so different scientists will call them different things. Sometimes, historically, we didn't even know these things. Two groups would be working on these things and not even know the other group existed, be working on it for 10 years, and then suddenly somebody reads the literature and goes, hey, these people are talking about the same thing and they're calling it two different things. You know, and they've been working on it for 10 years. So it's really strange. There's just way too much literature out here. It's getting out of hand. It is not organized by any international body or anything. And so you get all this really bad terminology. Now here's something we haven't talked about, sharp and broad promoters. All right, so let me very quickly, or peaked. Peaked is sharp. Dispersed is the same thing as broad. And what that means is when you have a piece of DNA and you have a gene, how many possible transcription start sites do you have? Now, you're going like, wait, there's only one transcription start site. It's right here, right? No, afraid not. There are genes out there that kind of works like introns and exons. You can have multiple transcription start sites, and depending on what's going on, you can start at different places. And this they leave out of intro textbooks. And what we found is what's going on is there are multiple promoters, multiple places that actually the polymerase can stick. So if I stick here, oops, sorry, I do this one, all right? If I start here, in other words, this is RNA polymerase sticking. If it starts here, you do this. If it sticks here, you do this piece, and if it starts back here, you do this, you'd make this one, which reminds me a lot of our some things in the operon, but it's not the same. And so what that means is a gene may have multiple places that RNA polymerase can actually bind and functionally make an RNA. In other words, there's not just one core promoter. You end up with 3 core promoters, and that, if this is the nature of your gene, then you have what's called broad or dispersed promoter. All right, and you need to know these. This is a big problem. The computer systems, I don't know of any computer program that can allow for this. This can only be found by human eyes. All right, and so you need to find, if you have one that has multiple start sites or multiple places RNA polymerase can attach in your promoter region, your regulatory region, then what you're going to find is when you start doing wet lab results is you're going to find RNAs of a different length, and they're not going to be spliced, or maybe they are spliced, but they're going to have these pieces at the beginning. And at first, when we discovered this, we thought it was a unique type of splicing, and it's not. It's actually due to the transcription start site just starting further back, being further back, based on the fact that you can have RNA polymerase stick at multiple places. And so what causes this? Well, CPG islands. Let's say I have C's and G's here. Okay. Well, if I methylate this one, okay, there's our little, that's methylated. All right, well, now you can't attach here. If I methylate this one, well, now you can't attach here. If I methylate this one, well, you can't attach anywhere and the gene is off. So you can methylate the DNA, and realize methylation happens on both strands. And so that's why it doesn't matter also previously when I was telling you, you really do need to look at both strands for a CPG island, because both strands get methylated. It's not just one strand. So you have C's and G's on both. So CPG island is something that occurs across the double-stranded DNA on both sides. Looking at this, what you have here is, depending on where I start my methylation, if I methylate this one, but I leave these two open, then you have two possible places. But maybe I methylate both of these, well, that would mean that you only get the small one. And then I methylate maybe this one, maybe I leave these two, and then I get both of these. But then if I methylate all three, I turn the gene off. So this is just a way, so you could get this one and this one, but not this one if you just methylate this one. Then if you methylate these two, then you're not going to get that one. You're only going to get this one here if both of these are methylated. All right, so just keep that in mind. You need to understand, now a peaked or a sharp one is one that only has a single promoter, okay, or a single place that RNA polymerases that can bind and move forward. All right, that is a peaked or sharp. I prefer sharp to peaked. I also use the term broad instead of dispersed, but these are terms that are also used. In fact, there are a couple more even. These are just the two most common sets of terminology. So, focused, that's another one. Focused, known as peaked, single dominant peak, narrow peak, sharp peak. The reason we have these names is because they were working in flies, and they called it one thing. They were working in humans and in rats, and they were calling it something different. And then the people in plants had a whole nother one. So we ended up with three sets of terminology, so you have to be careful. Now here's what we need to know. The sharp ones has either a single start site or a cluster of start sites in a small region. So it's not even as simple as I said. So if you have a single start site or several start sites very close to one another, then you call it sharp. I know, that's, yeah, okay. Dispersed or broad, all right, means that you have multiple start sites and you never know which one's going to be the right one. They're dispersed over a large distance, and you usually get these with housekeeping genes. Now, what is a housekeeping gene? Housekeeping gene is a gene that you don't ever want to turn off. All right, and this kind of makes sense why you might do it. So you're not likely to ever methylate a housekeeping gene. So these dispersed ones are unlikely to come with a CPG island. So the broad ones, these are not the ones we're looking for. We're looking for these. So in other words, we're looking for one or one with a small cluster. All right, we are looking for a sharp. All of ours should be looking for a sharp promoter, not a dispersed one. So this will make it easier on you. I'm just explaining what I just said. Whereas a constitutive means all the ways on. right? And that's going to happen through the use of multiple weak start sites. So in other words, constitutive will be the one that we're not looking at. We're not looking at a housekeeping gene. We are looking one with where we're controlling the. This is less. In other words, this one does less regulation. A sharp promoter means that this is not a highly regulated gene. Probably not regulated by the environment much. It's on, it stays on, and never goes off, and you need it to be pretty much working at a steady state at any given time. So that's a sharp promoter. I'm sorry, did I just say that backwards? I think I said it backwards. Sharp promoters are house creating using broad promoters, all right? Yes. Whereas housekeeping genes, I'm sorry, are broad promoters. Sharp promoters, I did say it wrong. Sharp promoters are the ones we're looking for, all right, and they are highly regulated. Yes, this is written poorly too. Did I write this? I probably paraphrased this from some paper, and I did a bad job of paraphrasing it. But we will be looking for sharp promoters that are either one or a cluster of them that are very close. Usually no more than 3 or 4, very close together. So that would be what we're looking for. All right. Now, facts list. So here we're doing sharp promoters. These are the ones we're looking for. All right. We're not looking for these. It says high GC, and that's not true. It is high GC. You don't turn these off, so they're usually not. I'm going to have to take some time to explain this. All right. Actually, here's the piece that's wrong. High GC, but not high CPG. That's the wrong place. All right. Now, low GC, but high CPG. There we go. Yeah, I need to fix this. This is very confusing. All right, and I wrote it wrong here. All right, so the exact location is determined by a TATA. All right, if a TATA is present. All right, that's the issue. If the TATA is present. And there needs to be something called an INR sequence. Actually, the INR should almost always be there. It's there, you just may not be able to find it. The sequence changes a little. But this is the real thing that acts like the TATA. The thing that gets things going, it's the sequence that has to be there. The way they just talk about TATA in the intro books, really it should be the INR, because that's what actually attracts the first proteins to start getting in there to do the transcription. All right, so just keep that in mind. We're doing sharp promoters. There are core promoter elements that you'll be looking for. And they are these. So somebody needs to go through and find these. All right, if they're there. We have no idea with algae. They're there in plants. At least most of these are. We need, let's see, MTR, BRE. I don't know if BRE is in plants. But you can go through and look for these. These are things that we will need to find. So someone in your group will need to be assigned to look for these. Now here, this is in the notation where W and R, like R is purines. Oh, I've forgotten what W is. N can be any, of course, and Y is pyrimidines. So purines, AG, R, just to remind you, means it can be an A or a G. And I put this on the announcements just to remind you. You have that as a handout, but if you've forgotten. And of course, pyrimidines are Y. And that would be, if this is A and G, then these are C and T, or T. All right, so keep that in mind. So you'll need to look for these. All right, so a TATA box, we've done that before. An INR, here's the INR I was just telling you about. But there are others. We need to find those, and we'll need to also find this TCT, BRE, MTE, and DPE motifs. These are very common ones. So read through this. TATA-like elements and their promoters are generally AT-rich. I mean, you're going to see a lot of A's and T's around a TATA. INR containing, here it goes, TATA-less genes. So genes that don't have a TATA, all right, realize 46% human lack a TATA and an INR. Actually, it's more than that now. They know more genes, and there are quite a few. The majority of genes in humans, now that we're really got this going, we're finding don't have. The most common is the CPG island, and that's the one you should probably look for. Now that's in humans. I have no idea what you're going to find in algae, and especially EHUCs. All right, the INR, here's your, talking about the INR here, they give you one for flies. And so this is mainly fly here, but you probably need to go more by, and here they're doing focused, which that would be sharp, that would be the one we're looking for. But this is flies. Insects have done away with CPG islands and do something different. So you might want to be careful about using any of these sequences in flies, because we do a lot of research in flies, but gene regulation is probably one they need to find a different organism to study this in, because they don't use CPG islands. TATA, we already know about TATA. So read this, but we already know, essentially, this is talking more about flies. It's because we've studied flies a lot, so we know a lot about how their gene regulation works. But for medicine, where we're looking at humans and mammals and vertebrates, we do a lot of things different. All right, so here's your basic eukaryotic promoter, and I'll end this lecture. It's definitely going to be more than one. Here you have your INR. Notice INR is where you start copying more. The TATA box is before it, and here you have upstream sites where you have your transcription factors binding, or your enhancers and things like that will alter the efficiency of your RNA polymerase up here. So right here, regulate the efficiency of utilization of the minimal promoter. And here's your minimal promoter. And notice it has a TATA and an INR. You do not have to have a TATA. You do have to have an INR, though. All right, so here we're talking about that. And here are the sequences. Here is another motif that looks like they're not mentioning flies here. So I would say that C or T. I guess I gave you wrong. That's pyrimidine. That should be C or. Oh, look it up in the box. Don't trust this and don't trust me. Go look it up. What I put on the thing. Let's see. Just keep in mind that this is the sequence you probably need to use, because they're not mentioning flies. Now here are the ones you're most likely to find, though, not the TATA. You're going to find either a GC box, that's the most common, an octamer, or a cat box. All right, and we've already kind of talked about cat boxes. Who's to say that this is not? This is related to the one in prokaryotes, but some people will call both of them cat boxes. Whether or not they're actually related, like the TATA, we don't even know if the two TATAs are actually evolutionary related. We assume they are. We don't know that for sure. So you've got the cat box. The here, let me undo. Let's see, how do I get it? Undo. There we go. Oh, it's the same thing. So here we go. And we are doing RNA polymerase II. So cat box, GC box, octamer. Make sure you look at those as you move through. And here are the locations. Notice the GC box is quite a bit downstream. The cat box is also, the TATA is at -30. All right, so the TATA is very close to the proximal promoter. These are kind of out there in, maybe you call them, actually this is very close to the core promoter. These are more in the proximal promoter. All right, and other enhancers like 100,000 base pairs away. You're really far away. So this is a good one. This is a good one to read. All right, read that one. All right, M3 and M22. Look for these. Someone in your group needs to look for these. BRE. There's a BREU and a BRED, and they come on either side. You don't usually get both of them. I think you usually only get one on either side. Right, it is uncommon for the promoter to contain both of these. One is going to be upstream, that's the U. One is going to be downstream. So in other words, that's like, here's your transcription start site. Upstream would be before the transcription start, and one would be downstream. So and they're slightly different. So you'll need to look for the BRE. Here is the sequence for the BRE. So it's a G or a C, followed a G or a C, G or an A, and a CGCC. And if you'll recognize this, this sure does look like a very similar sequence to what? Your GC box. And so look for the BREs. Realize you may or may not have a TATA, and one upstream and one's downstream. You usually don't get both. DPE, this is something that you may or may not find, but you need to look for. This is motif 10, and this is in Drosophila. Not sure you're going to find that, but go look for it anyhow. Let's see, do we have the sequence for it? Yes, we do. So look for it. So somebody needs to look for each of these sequences. I will let you guys break this up amongst yourselves. I don't think you're going to find the MTE and the DPE, but you might find something close. So work on it. You never know what plants have done. I'm not as well versed in plants as I am in humans and flies. The DRE, another Drosophila related one. So look for it, but you have no idea if you're going to find it. Here's your sequence to look for. And I do like it when I find things that tell you, okay, 5' to 3', so 5' side, that's always good. The DRE, here we go. And you can look for this one. See if you can find one or something very close. These continue on to that one, and so the X may be a member of a larger family. Activators in CPG islands. So this is something we will probably find because this is in CPG islands. All right, so here's one. You'll have to go decode this using the announcements where I put down the IUPAC nucleic acid symbols. All right, so look for those. And then here's something called the pause. The pause is something, it's an element that when a protein binds to it, it pauses the transcription. It causes it to pause for a little while, and something else will have to come along and let it go again. Now this is related also to RNA interference. I don't think we're going to get to this, but pausing can be caused by RNAs. And where do these RNAs come from? They're the introns that are spliced out often. All right, so you have exons and introns, you splice out one, and there'll be some gene that you want to pause until the other one is done, until the other protein is made, you don't want to start making the other one. So the intron gets spliced out and goes and does something very similar to this pause protein. So there are proteins that attach that pause, but there are also RNAs that pause. And they're usually, these little RNAs are the result of splicing of another gene. And the whole idea is I have two genes here, and what I want to do is, if I make this one, if I make the RNA here, I'm going to splice out a piece of it right here, and that's going to go attach in front of this one and cause this one to not be made while this one is being made. So I end up making this protein, alright, and what this does is it waits until you're finished making that, alright, then you make this one, alright, because this pause is just a pause, it doesn't last forever, and then you make this protein. So that way they have a sequence. So the intron does have a function. The intron can be used to pause other genes, alright, and so you pause the others so that they can come in the correct order. And this is kind of like the same function of your Z, Y, and A. You want these to be made in a particular order or a different amount or something like that, and you can have the RNAs and the introns go and regulate this. Really cool kind of stuff on this, but very confusing to people in a wet lab. A very difficult type of research to do. But of course this is actually pretty easy once you suspect that this is the case. Once you know that this intron right here, you just look for what it will stick to in front. But we're not talking about this right now. I'm telling you about it. I usually go into it more, but with this being online, this takes some amount of very complicated explanation that really, I have to say, has to be in person. But I wanted you to know about it. But we do have proteins that do the same thing. So there is a protein that can cause pausing, and it's called the DPE. And here we go. Let's see. Here's GAGA, also called GAF. All right. That's the protein, GAF or GRAF. It actually can be both. I looked that up the other day. So read this, know what's going on here, and I've already told you what GAGA can do. You don't know about these other things, but you can throw this in with your knowledge on the other things. And we have talked about this, so here's broad and dispersed transcription. All right, and here's some of this just talking about things. We don't have any sequences for you to look for, but they will tell you where to look for things. TCT, this is one that was mentioned before. We now have a sequence for that. Now, +1, this occurs right on the transcription start site. So realize this is something that binds directly on the transcription start site. That's what +1 means. So here is your TSS. You've got TY, TYY, and YT on either side. So this is not always what happens, but the problem is this isn't going to help you much because this occurs in RNA polymerase 1 and RNA polymerase 3 systems. You don't see it much in RNA polymerase 2, which is the one we are doing. But look for it anyhow. I don't think any of your genes are going to end up using RNA polymerase 1 or 3. These are special polymerases for making special types of proteins like the ones in the ribosome or for tRNAs and things like that. Alright, we've gone through CPG islands. I think I will start with this right here on the next video, and we'll end this video here.